All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. My apologies for the confusion that I said we won't have a talk and now we have a talk. Here is the talk, and I think this is one of the most important talks in for long COVID and for vaccine injured patients as well. This is a research from Italy, and researchers were trying to figure out the phenotype or the dysregulation of in the immune system which can be found in the long COVID patients. That was their question. What they found was this. So I'll tell you the summary now and then we'll go in the detail a little more. What they found was that in the long haul patients, interleukin 1 beta, interleukin 1 RA or receptor antagonist, interleukin 8, cytotoxic T cells or CD8 positive T cells were exhausted, helper T cells were exhausted. Cytotoxic T cells had a lot of PD-1 molecule expressing on them even when the patient had recovered. This is important. The patient had long COVID, but they had recovered from acute COVID, but their immune system was still dysregulated and not normalized. So the question in our mind should be, was this normalization supposed to happen? Was it supposed to happen that the CD8 cells that had PD-1 on them, PD-1 programmed cell death receptor 1 on their surface, imagine here, was it supposed to, were these PD-1 supposed to go away? Yes. In a normal infection process or even cancer process, when the immune cells become active, with their activity, they express a lot of PD-1 on their surface, which is sort of a brake, as we discussed yesterday. And if you press that brake, that would cause this immune cell to become stopped. However, by the end of the infection, these cells that have PD-1 do not express PD-1 anymore. So they become normal cells again. But in long COVID patients, cells with PD-1 stayed on. I would suspect a similar phenotype will be observed in vaccine injured as well. And the researchers saw that there was this presence of abnormal cells or dysregulated cells, and these interleukins coincided with long COVID symptoms. And then they took those cells out of the patients, long COVID patients, patients who were supposed to have recovered, but they still had the symptoms. They took the cells out of them, put them in a separate plate somewhere, and they did experiments on them by blocking their PD-1. And these same exhausted, dysregulated cells started functioning correctly. What does that mean? For physicians, if you have a patient of COVID long haul or vaccine injury, it may be useful to read these studies to figure out, number one, if the patient has interleukin 1 beta, interleukin 1 RA, interleukin 8 levels high, number one. Number two, it will be useful to see if their PD-1 expression is more, the cells, and if their PD-1, PD ligand, which are the molecules that bind with the PD-1, that level is more, and if their T cells, cytotoxic T cells especially, are exhausted. If that is the case, it is possible that such patients could be they might benefit from PD-1 blockers. So that is for the physicians to think about. Now let's start the discussion. Beautiful study and a newer understanding. So again, this is not the whole answer. the pathologies are varying. So not everyone would have this dysregulation, but some of the patients have this. So let's start. So this is drbeen.com. This is the study. Study says PD-1 blockade counteracts post-COVID-19 immune abnormalities and stimulates the anti-SARS-CoV-2 immune response. Stimulates the anti-SARS-CoV-2 immune response. Why do they say anti-SARS-CoV-2? And you could say, well, I do not need anti-SARS-CoV-2 response anymore. Their point was that when they tested the cells, these were exhausted cells. They were not reacting to SARS-CoV-2 antigens anymore. Then they corrected them by blocking their PD-1s. Then they have them attack the SARS-CoV-2 once more, antigen spike proteins, and they were able to attack. That means they restored them. So now it's not necessary that we only restore them for for SARS-CoV-2. They are restored normally to behave normally for other things too. So that is a study. There are some tables here in this study that are interesting for physicians. Now, the patients who were long COVID in this study, these patients had symptoms like the following: asthenia, that means lack of energy. You don't want to do anything, you just don't have energy. Dyspnea, breathing issues. I shouldn't have clicked there. Tachypnea, breathing, fast breathing. Cough, memory loss, chest pain, myalgia, muscle pains, erythema, redness, and nephropathy, kidney issues. These were the patients who had these, and they took their cells out, which were abnormal, and they were able to restore their cells. This is an ex-vivo study. And one more concept before we look at the study a little more: the concept of microRNAs. So I'm sure that we all now have heard of messenger RNA or mRNA. Vaccines are that way. Messenger RNAs are the little recipes that are copies of the genetic code in our DNA. So inside a nucleus, cell's nucleus, is a DNA. DNA has the recipe for the whole life of us, whole function, whole way we are built and functioning. The messenger RNA is sort of a photocopy of a page of that DNA, the big book of life. MicroRNA is a very interesting thing. It is another small sequence of genetic material, normally 22 nucleotides, very small. If you insert this genetic material in a messenger RNA, it silences it. A messenger RNA that has a microRNA inserted in it, that messenger RNA becomes useless. So if in a person, if in their immune cells, a microRNA against PD-1 is created, then their PD-1 will not function, the messenger RNA will not function correctly. It will get dysregulated. So the concept of microRNA, we'll talk more about it, but just keep in mind. And then here are some PD-1 blockers. PD-1 blockers are mostly used in cancers, and we talked about colorectal cancer yesterday. So PD-1 blockers have been out there for some time. Now those long haul patients or those severely vaccine injured patients, their doctors may be able to discuss this PD-1 with their trial teams, hospitals, and the patients. Okay, so now let's look at my drawings of this all. So these are our gifts for humanity. Today's gift was almost postponed. So study comes from Milan, Italy. Patients who were participants were from March 31st, 2020 to October 2nd, 2020. And the study is published in December 2021, so about 6 months ago. So it's a relatively new study. What did they find? They found in the long COVID patients, immune dysregulation. So they took, here is what they did. It's really interesting. They took cells from healthy donors, 15 healthy donors. They took cells from acute COVID patients, 30. And they took cells from post COVID people, they should not be patients, who had long COVID symptoms, 15. And now you would think that a person who has COVID and then they become recovered, then all COVID related changes in the immune system will become normal. And they found that in the long COVID, that was not the case. So they found that the T cells, especially cytotoxic T cells, the ones that are actually the fighters, the effectors, these T cells were PD-1 positive. I said that yesterday as well. I'll say it again. When a immune cell becomes active, at the time of activation, the immune cell expresses a lot of PD-1. What is the purpose of expressing them? The purpose is that, hey, if you want to stop me by stimulating PD-1. And as the cell's activity is not needed, as they take care of the infection, then the PD-1 expression goes away back towards normal. And the newer cells that are produced have almost no PD-1 or the normal PD-1 levels. Here they found that the cells had more PD-1s than normal, than needed. So why were there cells with the PD-1 expressed on them when there was no need for them to have PD-1? That was a dysregulation. They also saw interleukin 1 beta to be present in the plasma of the long COVID patients. They found interleukin 1 RA, which is interleukin 1 receptor antagonist interleukin, and they found more interleukin 8. So why do I keep repeating this? There are so many patients, there are so many people who say, what are the phenotypes? What are the things to measure? And there are so many doctors who are asking patients to go this measurement and this panel and that panel. In my opinion, autoantibodies panel, regular electrolytes, blood, complete blood counts, and these some inflammatory markers, with these markers is sufficient. Now, here is once again, this is the diagram to say that the immune system cells expressed a lot of PD-1. That means they were active. And if they keep expressing a lot of PD-1, and if you keep using their PD-1, then they'll be all exhausted as well. PD-1 is to exhaust them. Okay, then what did they see? They saw that ex vivo behavior of the T cell. So they took the T cells out. The behavior of the T cells was very interesting. So imagine if I'm long COVID, you take my cytotoxic T cells, you put them in a little dish, petri dish, experimental area. You have them attack SARS-CoV-2 antigen. This is a fake particle they made with the spikes on it. They put this fake particle with these cells. What should have happened? What should have happened is that these cells should have just attacked this as a normal behavior. Instead, these cells did not attack it. They were exhausted. They were not functioning correctly. And they had a lot of PD-1s in them. These were cytotoxic CD8 positive cells with the CD4 positive helper cells also doing this, but mostly CD8 positive cells, which were showing a lot of PD-1 on them as well. In addition to SARS-CoV-2 antigen, they actually exposed these cells to flu virus, to tetanus antigen, and other antigens as well. They kind of tried other antigens too to see if they would work at all, and these cells were not functioning. That means this patient's immune system was dysregulated for SARS-CoV-2, and not only for SARS-CoV-2, but for others as well. Why is that important? Look, when we have a T cell, every single T cell only responds to a specific antigen. One T cell or a B cell will not respond to antigen A, let's say flu virus and SARS-CoV-2 virus and Coxsackie virus and some EBV virus. It won't do that. One cell can only attack one part of the antigen. So, what they wanted to understand was, are these cells only exhausted for SARS-CoV-2, but they are okay for other antigens? And they found out that the T cells that they got out were not responding to any of the antigens. The immune system was generally dysregulated. That was bad. And that is bad because we still have long COVID patients. So, what did they find more? They found in the cytotoxic T cells, the effector cells, these are the cells that kill the infected cells or SARS-CoV-2 infected or bacterial or fungal infection or cancerous cells. Their function is to kill. They found that these CD8 positive cells or CTL cells had, see here first, let me zoom this in. So here is the, let's say, nucleus. Here is the nucleus. Within the nucleus, let's say this is the DNA of the cell. Within the DNA, for every gene, there is a promoter attached to the gene. Promoter is kind of a beginning, it is present at the beginning of a gene and it helps the gene to express. Imagine just like if you go to a hotel and there is a person who's standing at the door who would open the door for you. Promoters are usually the areas where enzymes come and attach, and this promoter area would help promote expressing this gene. They saw that the PD-1 promoter was methylated. There was activity there. Plus, the messenger RNA for PD-1 that was produced had microRNAs in that, which were microRNA 15-5P, which meant PD-1 function will not be functioning correctly. Dysregulation was present. In addition to the dysregulated cytotoxic T cell, I had not seen this happening. I was not suspecting this happening. I knew that there is immune system dysregulation. I was focusing on innate arm cells like natural killer cells, dendritic cells, macrophages. Then I was focusing on incorrectly produced memory cells that are just keeping the immune system, running. I was not suspecting the cytotoxic T cells themselves becoming dysregulated. And so when I had been thinking about long COVID and vaccine injury protocols, I never thought about this approach. So with that confession, let's go back here. In the plasma of these patients, again, long COVID patients, there were PD-L1 levels that were abnormal as well. So what is PD-L1? So PD, programmed death molecule, that is a receptor on the surface of the immune cell. The thing that attaches here, the other molecules that would come and attach here to stimulate it, these will be called PD ligand. And they are of two types, PD-L1 and PD-L2. For the medical professionals here or medical students here, nursing students, nursing professionals, PD-L1 are expressed on the lymphocyte and dendritic cells, which are outside of the bone marrow, mostly in the lymph node. PD-L2 are expressed on the bone marrow dendritic cells or bone marrow antigen presenting cells. So that's the basic difference. But anyways, PD-L1 and PD-L2 are the ligands that would attach with the PD-1 molecule and exhaust or stop the immune cells. So they saw in these patients' plasma, they were recovered patients. These patients were not supposed to have PD-L1s in them or PD-L2s in them above and beyond the normal ranges, but they had them. So their cytotoxic T cells were dysregulated, their genetic expressions were dysregulated, their mRNAs were incorrectly silenced, and these plasmas had a lot of PD-L1. So these cells were exhausted. They were not going to attack anything, may that be SARS-CoV-2 or others. I mean, we would have suspected that only SARS-CoV-2 antigen would be exhausted because these cells were reacting to them. But turned out that even other cells were exhausted in this whole process. And this tells us why other infections are also going incorrectly or are getting managed incorrectly. And I know some patients whose EB1 or Epstein-Barr virus is just so strangely continuing to not go away, just continuing to be present. And they had this. Anyways, back here. For the summary of this, what did they see? This is naive T cell. Right now in front of us is the acquired arm of the immune system, naive T cell. Naive T cell can become T helper 2 or T helper 1. T helper 2 can then cause B cell activation, and that would make antibodies. T helper 1 can activate the cytotoxic T cell. They actually saw all of them had dysregulation. They were all exhausted. The two issues. Number one, a majority of the immune cell type were exhausted. Number two, cells were exhausted not just for the SARS-CoV-2, but for other things too. They were not responding well to flu vaccine. That means if that patient gets flu, the patient is going to react abnormally to flu. These were not responding well to diphtheria and tetanus and so on. That was bad, is bad. Then what they did, so thanks to these researchers, what did they do? They actually brought these cells out in the little petri dish and they added PD-1 blockers. So this is what is called ex-vivo. Ex-vivo is where you take cells from the living organism, but you bring it out into an environment that is as similar as you can to the living, but the cells are coming from the living organism. This is ex-vivo. So kind of in vitro, but the cells coming from the actual patients. So in the ex-vivo setting, they blocked the PD-1 on these cells. Remember, these cells, PD-1 is dysregulated, so they blocked them. As soon as they blocked them, the cells became nearly restored to normal. That's the word they used, nearly restored. The cells became nearly restored back to normal. Can you imagine? Yes, I'm amazed at this. So their naive T cells started working correctly, helper cells started working correctly, their cytotoxic T cells started working correctly. All of a sudden, these cells were now attacking SARS-CoV-2 antigen. All of a sudden, these cells were now attacking flu vaccine. All of a sudden, these cells were attacking diphtheria, tetanus like things. The same cells. So the authors say that ex-vivo PD-1 blockade restored T cell response to SARS-CoV-2 antigen, immune checkpoint blockade. So PD-1 blockade is called immune checkpoint blockade because PD-1 functionality is called immune checkpoint. Immune checkpoint blockade ameliorates, this is their statement, ameliorates immune abnormality and stimulates anti-SARS-CoV-2 action. And this is my part, in exhausted immune cells. They said dysregulated cells. Here is a here is a cytotoxic T cell that has these PD-1s on that, and these little red things are the antibodies that are blocking the PD-1s. And as soon as PD-1 was blocked, the cell woke up and became ready, and it is now attacking the spike proteins. Can you imagine this happening? This can happen with in my opinion, this is not part of the study. This can happen with the vaccine too. So this will be, for severely injured patient, this will be another very important avenue to check. So this is it. This is the discussion. 26 minutes into it. I think I've even repeated it once. So you can, if you send it to someone to see it. You can say, hey, first 10, 15 minutes or first 26 minutes. Okay, now very quickly, this study is actually very, very detailed. And they have done a lot of work and lot of tests. Some things that I wanted to show you here is, for example, look at this. CD8 positive T cells, CD4 positive cells, CD8 exhaustion markers. And here, those, if you see in this p-value column, the rightmost column, wherever you see p-value that is significant, ns means not significant, so leave them. The significant parts is where they found the abnormalities in the immune system. For example, they found here OX40 positive that was significantly dysregulated. GITR positive on the cytotoxic T cells were significantly dysregulated. Why are these important? These are important for doctors and nurses and NPs and PAs and students to understand which kind of markers may be interesting. I already showed you these dysregulations. Now I wanted to show you something which is interesting. Here they said, overall, our results suggest an abnormal T cell phenotype, and function of the phenotype means a particular functioning state of a cell. An abnormal T cell phenotype and function of the immune response in patients who recovered from COVID-19. See, they're saying in patients who recovered from COVID-19, that means their immune system should have gone back to normal. But they are not really recovered. Who recovered from COVID-19, and this is at least partially due to dysregulated PD-1, PD-L1 axis in T cells. Interestingly, these immune abnormalities were found to be associated with a persistence of dyspnea and several additional COVID-19 related symptoms at the time of recruitment. This is the key statement here, that this dysregulation was associated with symptoms. So, lot more in this paper. I had to read it once, twice, thrice, and almost highlight everything to understand it. If you like me to go over the whole paper, please tell me. It would take more than an hour to do it. But I wanted to bring to you the basic gist of the paper and the clinical so what. They actually say it. This, if I look for the word clinical, here, a method to elicit a successful reversal of post-COVID-19 immune abnormalities may be necessary as long-term COVID-19 symptoms cause, in some cases, serious afflictions. We know that. And this strategy, the PD-1 blockade, could therefore be significantly clinically relevant. So, very important study. The way to look at these studies, or the way to kind of analyze these studies, is unfortunately, long COVID and vaccine injury is not a result of one abnormality. It is a result of multiple phenotypes or multiple pathologies, and everyone has slightly different combination of issues. At least for me, and you can call this as my ignorance, at least for me, I was not aware, I was not even suspecting PD-1 branch of the immune system, or that activity, that axis we call it, PD-1, PD-L1 axis, to be also the contributory factor. At least I got very much hung on to, number one, Dr. Bruce Patterson's discussions, then the immune cell shape issues, then the dysregulated macrophages, dysregulated mast cells, the memory cells and their abnormalities and presence of the memory cell in the wrong places, and the cells carrying the spike proteins. And I did not think about this side. And so here we are, another avenue. And why would I discuss this? Those patients who are becoming sick of their life because of the abnormalities and these symptoms, at least there is one more avenue to try for the doctors. So, with this, thank you very much. Please like, subscribe, and share. You may have noticed that I have stopped saying upfront to buy Dr. Bean plan if you like, but let me do it at the end. Felt it looked not nice of me to try to sell the product. Here is drbeen.com, and in the description of this video there is a link. The price is really low. This price would not be there. $67 for the lifetime access to 800 more lectures on Dr. Bean, and one time payment. It's not even recurring payment. So if you would like to support this work, you can become a member on Dr. Bean. In addition to that, there are links in the description. You can buy me a coffee, you can use PayPal, you can become part of Substack, you can become a patron, and so on. And least of all, you can do a like, subscribe, and share. And even in there, if you just want it to be really, really selective, then like and subscribe. So thank you very much. I think this should be shared because there are so many people who are suffering, and it is useful to share it with them. I feel shy asking for these things, but I think it should be shared. So with this, thank you very much. And let's see each other on the chit chat side, on the Cool Beans Cafe Live channel. I'll see you there. Bye.